This is an extension of a presentation given to a Roche-sponsored symposium during the Regional International Society for Blood Transfusion Congress in Perth, Australia, on the 29th of October 2025. In this symposium, I was honored to form part of a faculty with eminent blood safety experts Professor Helen Fadi and Dr. Paula Sa. I was tasked with reviewing the issues involved in establishing a plasma program for the provision of plasma-derived medicinal products, PDMPs, particularly in countries facing economic challenges. The global market for PDMPs has grown nearly sevenfold over the past 30 years, as shown by the slide showing data from the Market Research Bureau, an agency which collates, analyzes and reports on data from the plasma industry. Again, from the Market Research Bureau, we note that the global collection of plasma for fractionation also continues to grow briskly, driven primarily through the collection of plasma by apheresis of the paid donor population of the United States. Following a drop in collection during the COVID-19 pandemic, the sector recovered quickly and now totals over 90 million liters of plasma collected annually. Plasma recovered as a byproduct of voluntary blood donation, particularly in Europe, has grown as well, but at much smaller rates. The collection of blood and plasma is not uniform globally and is a function of the economic status of the countries involved. As this data from the World Health Organization shows, collection in high and medium income countries, as defined by the World Bank, is much higher than in low and low middle income countries. And this situation is shown in this map of the blood need to supply ratio of the different countries of the world. A low ratio indicates a high capacity to supply a country's needs, and it is clear that the low-income countries are experiencing difficulties in meeting their needs relative to the high-income countries. Clearly, all this confirms the role of economic status in determining the collection and provision of blood, as shown on this slide. High-income countries provide much higher levels of funding for their blood systems compared to low and lower middle-income countries. And as well, with apheresis collection, which is more expensive than whole blood collection, this is limited to the upper and high-income countries. And low-income countries have still not covered their have not converted their blood donor populations to a voluntary, non-remunerated base, indicating a vulnerability of the blood systems compared to higher income countries. This is also reflected in the proportion of red cell transfusions given as whole blood, which is much higher in low income compared to high income countries. This is an important point, which I will address further momentarily. It is important to assess some aspects of blood safety, which are important for all blood-derived therapies. I draw attention to the dynamics of viral replication in infected individuals, and in particular to the serologically silent window period of viral replication, which is undetected in donations which are screened solely with immunological markers, but which can be detected with much higher sensitivity by nucleic acid testing, NAT. This is shown on this very familiar representation on this slide. It is important to note that the prevalence of viral markers detected by screening tests is also highly dependent on the income status of countries, with low-income countries showing higher prevalence throughout. In order to improve this situation, while avoiding the high expense of NAT, many low and middle income countries have introduced other forms of testing for addressing the serological window period. Many of these types of tests are less sensitive than NAT, with a lower capacity to shorten the window period. This has implications for the safety of blood and plasma products derived from such donations. For plasma products derived from manufacturing pools of many thousands of individual plasma donations, the introduction of donations which are positive because of absent or inadequate testing has profound effects on the safety of the manufactured products. In the absence of adequate viral inactivation steps in the manufacture, these products will inevitably expose patients to the infectious agents. This is shown in this estimation, which relates the prevalence of the infectious agent, the scale of manufacture, and the number of treatments to yield a figure for the exposure of the agent. As can be seen, at the usual scale of manufacture and with a relatively high prevalence agent, exposure is inevitable. This has been avoided for the past 30 years by introducing steps in the manufacture which are designed and validated to inactivate and remove contaminating pathogens such as viruses. The lack of PDMPs in the low and middle income countries has severe consequences for patients. This occurs because PDMPs are unavailable, unaffordable, or available in insufficient quantities. Frequently, there exists a lack of understanding on the clinical value and the use of PDMPs. All this affects mostly patients with rare disorders such as the hemophilias and primary immune deficiencies, and there is an important role for patient organizations with these disorders in asking for better access. A lack of access to safe PDMPs leads to patient morbidities associated with high societal costs, and while humanitarian aid programs, such as those of the World Federation of Hemophilia, are valuable to help to meet increasing and diversified therapeutic needs, they represent an intermediary but not a sustainable solution. An example of this issue will be offered later in this talk. It is useful to envisage the path towards the provision of PDMPs by national blood systems as occurring through a series of sequential phases, evolving from the basic provision of blood for transfusion to the provision of specific components from this blood and the consequent generation of a supply of plasma surplus to clinical need and thus available for conversion into PDMPs. This may be supplemented according to affordability with plasmapheresis as a source of plasma dedicated solely for manufacture. This plasma supply is best converted into PDMPs via fractionation by a licensed manufacturer through suitable contracts. Once economic and other factors permit, consideration may also be given to the construction of a plasma fractionation plant, which may be national or geared towards regional needs. I will offer some comments on the first step, the provision of blood for hospital use. In this graphic from the WHO, we see the demographic and clinical use in the transfused populations of Benin, a low, middle and income country, and Denmark, a high-income country. In Benin, the transfused population is much younger than in Denmark, and most blood is transfused for acute rather than chronic indications. It is important that products are geared to clinical need, and the extinction of whole blood as a transfusion modality, as is found in high-income countries, would harm patients in countries such as Benin. It should be noted that even in high-income countries, the benefit of targeted whole blood transfusion has been recognized in these past years, and whole blood has made a return to the transfusion armamentarium of these countries as well. The issue of humanitarian aid in the establishment of blood systems was assessed some years ago by a group of eminent transfusionists in this paper from PLOS. It was shown that sub-Saharan African countries, which, funded by the United States President's Emergency Plan for AIDS Relief, known as PEPFAR, which was established by President George W. Bush, had converted their blood systems to Western norms and had eliminated, as a result, whole blood as a therapeutic modality. This increased considerably the cost of transfusion for health insurance purposes and also generated a considerable volume of fresh frozen plasma, which, being in excess of local clinical needs, was largely discarded. We shall return to this issue momentarily. Continuing with the progression of PDMP provision, we approach the stage of producing components according to the needs of the health system. Clearly, this is also dependent on economic status, with relatively low levels of component production in the low-income countries. I reiterate that this is not necessarily a bad thing if it is reflective of the clinical needs of the health system at that point in time. However, as stated previously, much of the recovered plasma is discarded, and in Africa, this situation is particularly visible through this report from the WHO, addressing most of the blood collected in Africa. Nearly half of the red cells were transfused as whole blood, and less than half the donations collected were processed into components. The plasma thus generated cannot be used clinically because it is not indicated in most of the clinical milieu of these countries, and much of it cannot be channeled into fractionation because of lack of quality and safety and because of inadequate financial resources. The establishment of good manufacturing practices is required by fractionators who may be available for fractionating this plasma, and this is frequently lacking in low-income country blood centers. In addition, the lack of sufficiently sensitive infectious screening tests is also an impediment. If these problems can be overcome and the plasma flow of adequate safety and quality can be generated, this can be channeled into fractionation. This requires the establishment of appropriate contracts to manage the process and ensure the return of the PDMPs to the country of plasma origin. Before this is undergone, however, an assessment of the national clinical needs for PDMPs is required. As was shown previously for blood components, the clinical needs profile for countries of different economic status varies considerably. This comparison between Indonesia and France demonstrates this. In low and middle income countries, albumin is generally still a major product, needed clinically at higher levels than for high income countries. And financial constraints limit the provision of recombinant coagulation factors, which have replaced plasma-derived products in the high-income countries. These differences are very clear when looking at the situation in Indonesia and France. When establishing a contract fractionation agreement, these considerations are important. Plasma quality, including donor screening, donation testing, processing, and all other features, must comply with the fractionator specifications and the regulatory requirements. This could be compiled into a plasma master file, as I describe in another talk. Audits of the blood establishment and the blood collection practices should be audited by the plasma fractionator. The licensing of the blood establishment by the local regulatory authority should be ascertained, and for the collection of plasma for fractionation. And this should also be ensured for the licensing of the contract fractionator and the resulting fractionated products by the local national regulatory agency. Such an agreement, when well constructed and monitored, has several advantages. It is a good route to domestic plasma and to avoid wastage, while guaranteeing some stability in the supply of PDMPs. It contributes to the sustainability of blood establishments and ensures the optimal use of plasma. Agreements should involve a learning phase, establishing quality criteria of plasma for fractionation with progressive GMP improvements at the blood establishment, which will then benefit all blood components. In addition, training on plasma fractionation can be imparted prior to the potential technology transfer for fractionation with a domestic plant. The system for the generation of plasma products from plasma of different origins comprises several contributors, which may interact in different ways. These contributors include domestically sourced plasma, which may be fractionated in the country of the plasma's origin to plasma-derived products for patients in that country of origin. The domestic plasma may also be sold to fractionators acting within and outside the borders of the plasma's country of origin, while plasma-derived medicines produced from diverse countries and independently from the domestic plasma are sold to the public health system. Finally, mixed models would comprise domestic plasma fractionated under contract to one or several fractionators being used to produce plasma-derived products, with the open market drawing on products from several sources, what is used to augment the supply of domestically sourced products. The first type of model, where the domestic plasma is fractionated within the borders of a country and provides all that country's plasma products, is not found in the continent of Europe. It is arguable whether it is found anywhere in the world, as the United States is totally self-sufficient in its needs for plasma derivatives, but fractionation is also done outside the border. Cuba has a system of self-sufficiency in plasma and plasma products, developed as a result of economic restrictions. It is not known if this system meets all of Cuba's needs. Some countries in the European Union are now embracing the second type of model, whereby their domestic plasma is sold to fractionators without any linkage of the resulting products to the public health system. And these include the Czech Republic, Finland and Sweden, countries which source all their plasma product needs from the open market, while collecting considerable volumes of plasma which is sold to commercial companies. The mixed model is used by a range of countries and is shown in the bottom part of this slide. These countries all commission fractionation companies to generate plasma products from domestic plasma, with the products returning to the public health system. A number of variants of this model of contract fractionation are found, whereby contracts are awarded under tender systems or directly to a single fractionator. Additional needs are met through purchase from the open market. And here we see programs in which a national fractionation plant produces PDMPs for healthcare systems, providing domestically sourced plasma from voluntary donors, and others in which such systems establish contract fractionation with a commercial fractionator, delivering PDMPs to the agencies generating the plasma. Several low and middle-income countries are emerging as players in these kind of arrangements. In cases where clinical needs for certain plasma products are still outside the clinical environment, surplus plasma can still be used to generate these products, which can then be commercialized to provide funds for the blood system's development. Such an arrangement has been described in Namibia, where plasma products fractionated by a South African not-for-profit company are partly commercialized, and the funds are used for paying for individual donor NAT, improving significantly the safety of blood components provided by the blood system. Note that this initiative was developed by the Namibia Blood Service after the cessation of PEPFAR funding left a gap in the budgetary sustainability for the blood system, emphasizing the transient nature of such humanitarian arrangements. Similarly, blood banks in India have been allowed through regulation to sell plasma from voluntary donors to the commercial fractionation sector. Several conditions are involved in this system, which are designed to optimize outcomes for both the blood supplier as well as the fractionation company. It is to be hoped that such arrangements improve the status of blood safety in India, which recent events have been shown to be inadequate in some regions. As we approach the final phase in the route to domestically sourced PDMPs, we consider the issues around the establishment of a national fractionation plant. It is important to appreciate the challenges in establishing such a facility, as shown by the large number of failures in this regard experienced in the past decades. However, a large country and populous regions have a legitimate aspiration for such a facility, and the ongoing process in Indonesia is of great importance and interest to us all. It is noteworthy that Indonesia has followed a staged path as described in this talk, with a designated external fractionator engaged to contract fractionate Indonesian plasma, while the same agency builds a plant for eventual use in Indonesia. And so I come to share my final thoughts. I reiterate that the first task for a national blood service is to ensure sufficiency in the supply of safe blood and components for hospital use. Once this is achieved, a plasma program may be considered. A number of models are available for the establishment of a plasma policy, and no size fits all. The safety and quality issues around plasma products require strict adherence to GMP and pathogen safety. The first step in assessing such a project should include a needs assessment, which should be based on the particular medical environment of the country in question. Contract fractionation with a reputable fractionator is the best route to PDMPs for low and middle income countries, but a domestic fractionation capacity is a legitimate aspiration for populous regions, in which case a cautious approach should be combined with a willingness to collaborate internationally as well. I draw attention to these publications, all of which are available on open access in Vox Sanguinis. These form the basis of much of the thinking in this presentation. And I thank my colleagues and teachers over the 45 years I have spent in this sector. In particular, the presentations use the public thoughts of my friend Professor Thierry Bernouve, and these are gratefully acknowledged. Here we are together, some 17 years ago, and I think we look as good now as we did then.